Hello everyone, hope all you are good in this situation. Myself Debojit Boydya Chaudhary, bearing roll number FEM 19017, Department of Food Engineering and Technology, Tezpur University, Assam. Today I am presenting on MAP Modified Atmosphere Packaging, its product safety and its nutritional quality. Now coming to the next slide, contents. The contents of my presentation contains what is MAP, typical gases for modified atmosphere packaging, benefits of MAP, food stuffs suited to modified atmosphere packaging, carbon dioxide as an antimicrobial gas, safety of MAP product, and then next effect of MAP on the nutritional quality of packaged food products, and then conclusion, and finally the references. So coming to the next slide, what is MAP? According to the Food and Drug Administration, modified atmosphere packaging involves either actively or passively controlling or modifying the atmosphere surrounding the product within a package made of various types and or combination of films. Or it can be defined as the enclosure of food product in gas barrier materials in which the gaseous environment has been changed. MAP enables the fresh produce or perishable product to be packaged when it is fresh and then maintains it in that condition. Thereby, it's reducing distribution cost and enhancing flavors and nutritional value for the consumer. Now the MAP packaging aims to provide longer shelf life, maintain sensory attributes like color, appearance, taste, and achieve the food safety of the product. The normal composition of air present in MAP is 21% oxygen, 78% nitrogen, and less than 0.1% carbon dioxide. Now MAP contains mainly used for plastic flexible laminates are polyethylene, polyamide, polyethylene terephthalate, polypropylene, polyvinyl chloride, ethylene vinyl alcohol, and polyvinyl phenylidene chloride. Now MAP are rigid, semi-rigid structures are commonly produced from polypropylene, then polyethylene terephthalate, and unplastic PVC and expanded polystyrene. Now moving to the next slide, it contains figure of modified atmosphere packaging in which various food products are packaged through modified atmosphere packaging. Here we can see the various food items which can be packed through modified atmosphere packaging. Now the next slide contains typical gases for modified atmospheric packaging. Typical gases contain oxygen, which is essentially cause food to spoil due to oxidation and forms the ideal preconditions for aerobic microorganisms to grow. As a result, oxygen is frequently excluded from modified atmosphere packaging. Now carbon dioxide, which is colorless, odorless and tasteless. The gas is frequently used to increase the shelf life of food. The shelf life of packaged or storage food is normally longer, the higher the CO2 content. Now nitrogen. Nitrogen is an inert gas that is used to expel air, especially oxygen, out of the packaging. It is also used as a filling gas, which equalizes the effect of carbon dioxide absorption by the perishable food. Now carbon monoxide, which is also colorless, odorless and tasteless. Similar to oxygen, carbon monoxide is sometimes used to retain the red color, primarily the meat. Now, argon. Argon, which is inert, colorless, odorless, and tasteless. It is to be believed that certain enzyme activities are inhibited and argon slows metabolic reaction in some types of vegetables. Now, hydrogen and helium, which feature in modified atmospheres in some application, more or less some application. However, these gases are not used to extend shelf life. They are used as trace gases for some leak detection system. These are basically used in leak detection of MAP packaging on the since these gases otherwise has no positive properties on the food products and are expensive and not easy to handle, and it is not basically used by normal people, and it is very used to rare. Now moving to the next slide, we can see there are various food products and their composition of carbon dioxide, nitrogen and oxygen. We can see from the table red meat contains 30% carbon dioxide, no nitrogen and 70% oxygen. And then we can also see fish, which is 40% carbon dioxide, 30% nitrogen and for 30% oxygen. And various other food items we can see from the table their composition of carbon dioxide, nitrogen and oxygen. Now moving to the next slide, which is benefits of modified atmosphere packaging. Now the first benefits are longer shelf life, higher quality, which means its shelf life increases from 50% to 400%. Second, less waste. It reduces economic losses due to longer shelf life. Third, more sales opportunities. For growers and consumers, it can extend the season of certain fruits and vegetables, fewer preservatives. In modified atmosphere packaging, enables processor to extend shelf life without using preservatives. It is also ideal for the packaging of organic produce. Now the next slide contains foodstuffs suited to modified atmosphere packaging. There are various foodstuffs which can be used for modified atmosphere packaging, like meat and sausage products, fish and seafood products, dairy products, bread and bakery products like bread, cake, pastry, fruits and vegetables, pasta and ready-made meals, wine, whiskey, and many more things. Now, coming to the next slide, as carbon dioxide as an antimicrobial gas. When carbon dioxide is added in package, it partly dissolved in the water and fat phase of food. Carbon dioxide in MAP results in an increased lag phase and generation time during the logarithmic phase of growth of the organism involved. The antimicrobial action of carbon dioxide is as follows. Number 1, alteration of cell membrane function including effects and its nutrient uptake and absorption. Now 2, direct inhibition of enzymes or decrease in the rate of enzyme reaction. Now number 3, it's penetrate into bacterial membranes leading to intracellular pH changes. And last, direct changes to the physiochemical properties of proteins. Now moving to the next slide, which contains safety of modified atmosphere packaged food and its product. Now here you can only go to the microbial safety of MAP. Here number 1, Clostridium botulinum Foodborne botulism is a severe illness that is caused by the consumption of foods containing a neurotoxin produced by a bacterium called Clostridium botulinum. The possibility is that psychotropic non-proteolytic strains of C. botulinum types B, E, and F are able to grow and produce toxins under MAP conditions. The psychotropic strains of C. botulinum are able to produce toxin in an environment with up to 10% oxygen. The probability of one spore of non-proteolytic Clostridium botulinum, that is types B, E, and F, being toxicogenic and rockfish. The result which shows that the toxicogenicity was significantly affected under the probability less than 0.005 by temperature and storage time, but not by the used modified atmosphere, which is vacuum 100% carbon dioxide or 70 to 70% carbon dioxide or 30% air. Now, toxin produced from non-proteolytic C. botulinum type B spores was also retarded by a carbon dioxide enriched atmosphere, which is 30% carbon dioxide or 70% nitrogen. Now moving to the next slide, the next microbial safety is Listeria's monocytogenes. Here it is Listeria's monocytogenes is a psychotropic foodborne pathogen. Here we can see the structure of Listeria monocytogenes from the figure. Now, Listeria monocytogenes is not greatly inhibited by carbon dioxide, which is enriched by atmosphere, even though when combined with other factors such as low temperature, decreased water activity, and also the addition of sodium lactate. The inhibiting effect of carbon dioxide is significant in this Listeria's monocytogenes. Now, Listeria growth in anaerobic carbon dioxide enriched atmosphere has been demonstrated in lamp in an atmosphere of 50 is to 50 carbon dioxide by nitrogen at 5 degree centigrade, and in pork in an atmosphere of 40 is to 60 carbon dioxide by nitrogen at 4 degree centigrade. These are the various examples. Now, Listeria monocytogenes fail to grow at 4 degree, 10 degree and 27 degree centigrade in an anaerobic atmosphere which is contain 75% carbon dioxide and 25% nitrogen. Listeria monocytogenes was also only minimally inhibited on chicken legs in an atmosphere containing 10% oxygen and 90% carbon dioxide. These are very few examples of Listeria monocytogenes which can be saved from MMP products. Here we can also see the structure of Listeria monocytogenes from the given figure. Now coming to the next slide, which is Yersinia enterocolytica. Yersinia enterocolytica is regarded as one of the most psychotropic foodborne pathogens, and the growth of this was described in vacuum packaged lamp at 0 degree centigrade. Carbon dioxide retards the growth of Yersinia enterocolytica at refrigerated temperatures. Oxygen also seems to play an inhibiting role on the growth of Yersinia enterocolytica. Now, the next bacteria is Aeromonas species. Arowana species are stored in refrigerated conditions. The growth of A. hydrophilia has been detected at low temperatures in a variety of vacuum packaged fresh products. The behavior of cocktail A. cavei and A. bacterium in air or in low oxygen, low carbon dioxide atmosphere was investigated in fresh cut and vegetables. Here from the slide we can see these two bacteria from the figure. These are very much effective. Now, what is Clostridium botulinum? Is a bacterium that produces dangerous toxin which under low oxygen conditions can affect MAP products. From this we can safety aspects has been taken. Now moving to the next bacteria, which is Listeria monocytogenes, is the species of pathogenic bacteria that causes the infection listeriosis. It is a facultative anaerobic bacterium capable in the surviving presence or absence of oxygen. From this we can also save our MAP products by doing many things. Next, Yersinia enterocolitica, which is gram-negative bacillus-shaped bacterium that causes a zoonotic disease called Yersiniosis, from which we can save our MAP product by doing many applications. Last one is Arowana species. It's a genus of gram-negative facultative anaerobic rod-shaped bacteria that morphologically resemble members of family, and most of the species are A. hydrophia, A. cavea and A. veroni fiber sobria. Now coming to the next slide, which is growth of Aromonas hydrophilia in defined atmosphere. Product type beef, sliced roast beef, lamb and their pH condition beef, which is less than 6, sliced roast beef 6.1, lamb 5.4 to 5.8, another lamb less than 6, and their temperature conditions beef minus 2 degrees centigrade, again 0 degrees centigrade, again 2 degrees centigrade, again 5 degrees centigrade, again 10 degrees centigrade. Sliced roast beef minus 1.5 degrees centigrade, 3 degrees centigrade, and their storage time are also shown in the table, and atmospheric condition, which is also shown their percentage by oxygen, carbon dioxide and nitrogen, and there also shows increased log phase CFU per gram, which is shown in the table. We can see from the table the growth of Aromonas hydrophilia in different atmospheric conditions. Now coming to the next slide, which is effect of MAP on the nutritional quality of packaged food product. Now the first, the effect of MAP on the nutritional quality of non-aspiring food products. What is non-respiring food products? In the case of non-respiring food products, the starting head-based atmosphere composition should be tailored on the basis of specific deterioration process and the interaction with headspace to guarantee the optimal preservation. Here, the shelf life of the packaged products can be extended by 50 to 200% by using modified atmosphere packaging. Very little information is available about the influence of modified atmosphere packaging on the nutritional quality of non-respiring food products. For packaging non-respiring food product, oxygen is excluded from the atmosphere and therefore one should expect a retardation of oxidative degradation reactions. There are bakery products, there are that are susceptible to spoilage by bacteria, yeast, and molds can have their shelf life extended by MAP with high carbon dioxide content, although cost limit this application. The most common forms of deterioration are microbial spoilage, staling and moisture loss or gain and use of carbon dioxide has an inhibitory effect on the growth of most microorganisms and is suitable for food preservation and stability, toxicity, organoleptic and economic grounds. To avoid packaging collapse problems encountered with high carbon dioxide content, mixtures of carbon dioxide and nitrogen are generally adopted. Now moving to the next slide, which is the effect of MAP on the nutritional quality of fresh fruits and vegetables. Here, the nutrient content of fresh fruits and vegetables can be influenced by various factors such as genetic and agronomic factors, maturity and harvesting methods, and post-harvest handling procedures. The fruits and vegetables are rich sources of micronutrients and dietary fiber. They also contain an immense variety of biologically active secondary metabolites that provides the plant with color, flavor, and sometimes anti-nutrition or toxic properties. Storage of fresh fruits and vegetables within the optimum range of low oxygen and elevated carbon dioxide atmosphere for each commodity reduces their respiration and CH4 for production rates. Optimum control atmosphere retards loss of chlorophyll, biosynthesis of carotenoids and anthocyanins, and biosynthesis and oxidation of phenolic compounds. In general, control atmosphere influences flavor quality by reducing loss of acidity, starch to sugar conversion, and biosynthesis of aroma volatiles, especially esters. Retention of ascorbic acid and other vitamins result in better nutritional quality and includes antioxidant activity of fruits and vegetables which keep in their optimum control atmosphere. However, little information is available on the effectiveness of control atmosphere on the nutrient retention during storage. The influence of C- control atmosphere or modified atmosphere on the antioxidant constituent related to nutritional quality of fruits and vegetables including vitamin C, carotenoids, phenolic compounds as well as glucosinolates will be seen here. Now the vitamin C. Vitamin C in fresh fruits and vegetables undergo changes for the moment of harvest, and since ascorbic acid is one of the more reactive compound, it is particularly vulnerable to treatment and storage conditions. In broad terms, the milder the treatment and lower the temperature, the better the retention of vitamin C. But there are several interacting factors which affect ascorbic. Vitamin C is one of the most important vitamins in fruits and vegetables for human nutrition. More than 90% of the vitamin C in human diets are supplied by intake of fresh fruits and vegetables. Vitamin C is required for the prevention of scurvy and maintenance of healthy skin, gum and vessels. Vitamin C is also required for this pandemic situation as it boosts the immunity of human. A reduction in temperature and of oxygen concentration in the storage atmosphere have been described as the two treatments which contribute to preserve vitamin C in fruits and vegetables, and others observed that loss of ascorbic acid can be reduced by storing apples in a reduced oxygen atmosphere. It's an example. In fresh cut products, high carbon dioxide concentration in the storage atmosphere has also been described to cause degradation of vitamin C. The results vary among fruit species and cultivars, but the tendency for reduced O2 and carbon dioxide level to enhance the retention of ascorbate. One more studies in which cut broccoli flores and intake heads of broccoli, MAP results in greater ascorbic acid retention and shelf life extension in contrast to air stored samples. In conclusion, the loss of vitamin C after harvest can be reduced by storing fruits and vegetables in atmosphere of reduced carbon dioxide and or up to 10% carbon dioxide. Control conditions or do not have a beneficial effect on vitamin C if high carbon dioxide concentration are involved. Now coming to the next slide, which is carotenoids. Carotenoids are form one of the most important classes of plant pigments and play a crucial role in defining the quality parameters of fruits and vegetables. The best documented and established function of some of the carotenoids is their provitamin A activity, especially of beta carotene, alpha carotene, and beta carotenoxanthin also possess provitamin A activity, but to a lesser extent than does carotene. Modified atmosphere includes either reduced oxygen or elevated carbon dioxide are generally considered to reduce the loss of provitamin A but also to inhibit the biosynthesis of carotenoids. Control atmosphere of 3% oxygen and 20% carbon dioxide both alone and together with ethylene prevented total carotenoid and lycopene biosynthesis on tomato. The carotene content of leeks was found to be higher after storage, 1% oxygen plus 10% carbon dioxide, than after storage in air. Carotenoids, their role in the plant is to act as accessory pigments for light harvest and the prevention of photo-oxidative damage as well as acting as attractants of pollinators. The given example, the in addition the content of beta-carotene in broccoli flowers increase at the end of control storage. Percentage of oxygen is 2% plus 6% carbon dioxide and remain stable after returning the sample to ambient conditions for 24 hours. Now moving to the next slide, which is phenolic compounds. Controlled atmosphere and modified atmosphere packaging can directly influence the phenolic composition as reflected in the change observed in anthocyanin. Carbon dioxide enriched atmosphere, which is less than 20%, used to reduce decay and extends the post-harvest life of strawberries, induced a remarkable decrease in anthocyanin content of internal tissues compared with the external ones. Very few evidence is found for the role of antioxidant constituents of fruits and vegetables in maintenance of health and disease prevention. There are many studies which shows that consumption of fruits and vegetables with high phenolic content correlates with reduced cardio and cerebrovascular disease and cancer mortality. Generally, an increase in phenolics is considered a positive attribute and enhances the nutritional value of plant products. However, many secondary metabolites typical of wild species of fruits or vegetables have been toxic effects although they are not considered. In addition, the organoleptic and nutritional characteristics of fruit and vegetables are strongly modified by the appearance of brown pigments. Modified atmosphere can also have a positive effect on phenolic related quality as in the case of prevention of browning of minimally processed. In addition, modified atmosphere packaging of minimally processed red lettuce 2 to 3% O2 plus 12 to 14% CO2 decreased the content of flavonol and anthocyanins of pigmented lettuce tissues when compared to air storage. Thus, the increase of soluble phenyl propanoids observed in midribs of minimally processed literacy after storage in air was avoided under MAP. There is another example which shows that there is decrease in all phenolic compounds. Example anthocyanins, flavanols and caffeol tartaric and p-coumaryl tartaric acid in both skin and pulp of grape berries rapidly brought under anaerobiosis in carbon dioxide enriched atmosphere. Now moving to the next slide, which is glucosinolates. Here brassica vegetables such as cabbage, Brussels sprouts, broccoli and cauliflower are an important dietary source for a group of secondary plant metabolites known as glucosinolates. The sulfur containing glucosinolates are present as glucosides and can be hydrolyzed by the endogenous plant enzyme myrosinase. Here, myrosinase and the glucosinolates are physically separated from each other in the plant cell, and therefore hydrolysis can only take place when cells are damaged. Here, indole 3 methylglucosinolates, which occur in appreciable amounts in several brassica vegetables, are of interest for their potential contribution of anti-carcinogenic compounds to the diet. The process, such as chopping, cooking, and freezing, influences the extent of hydrolysis of glucosinolates and the composition of final products. There are very few reports describing the effects of storage on the glucosinolate content. For instance, the storage of white and red cabbage for up to 5 months at 4 degrees centigrade, which does not seem to affect the levels of glucosinolates. However, there is little still information about the influence of modified atmosphere packaging on total or individual glucosinolate content of brassica vegetables, but an increase in total glucosinolate content was reported in broccoli florets when stored in air or controlled atmosphere while in absence of oxygen with a 20% carbon dioxide resulted in total loss. Now coming to the next slide, which is conclusion. Here I am concluding my presentation, which is modified atmosphere packaging is likely to be one of the important technology for packaging of fresh produce. The advantages of modified atmosphere packaging includes extending the shelf life, maintaining the quality of fresh produce, and suitable for use and marketing. There are various challenges in modified atmosphere packaging involved cost of packaging materials, storage temperature and specific gas consumption for specific product. Every technology has its own pros and cons. Hence, balance has to be achieved in such a way that can assure safety and quality. Modified atmosphere packaging films developed to control the humidity level as well as the gas composition in the sealed package are beneficial for the prolonged storage of fresh fruits, vegetables and herbs that are sensitive to nature. These films are commonly referred to as modified atmosphere or modified humidity packaging films. Modified atmosphere packaging does not slow the growth of some harmful bacteria and therefore is usually used in conjunction with other preservation techniques such as refrigeration. While modified atmosphere packaging can extend the product's shelf life, it does not eliminate all bacteria. Therefore, other preservation methods must be used. Bacteria continue to grow especially in warmer temperature, so refrigeration of products even after they have been packed with modified atmosphere packaging procedures is recommended. Therefore, the application of various additives and other preservation techniques in collaboration with modified atmosphere packaging is an area of research interest. Moving to the next slide, which is references. The references which I have taken for making this presentation are shown in this slide. Thank you. for watching my presentation. Now I want to say one line. The easiest way to become a superhero is now. Just stay at home, stay happy, stay alive. Thank you.